In Chapter 15, Different Emotions Activate Different Regions of the Human Brain, it says that several different forebrain areas are consistently implicated in varying emotions. For example, in Love versus Friendship, it showed increased activity in the insula and the anterior cingulate cortex, and subcortically in the caudate and putamen, all bilaterally. It also showed reduced activity in the posterior cingulate and amygdala and in the right prefrontal cortex. This combination of sites differ from those in other emotional states, which suggests that a unique network of brain areas is responsible for the emotion of love. In envy, there was increased activity of the anterior cingulate cortex activity. In a study done by Antonio Damasio, brain activation was compared during 4 different kinds of emotion, and the insula, cingulate cortex, and prefrontal cortex were the regions involved. Adults were asked to recall and attempt to re-experience episodes involving sadness, happiness, fear, or anger, as well as a neutral episode. The participant was asked to signal as soon as the desired emotion was experienced, and PET images of the brain activity were made. Physiological responses preceded the reaction as well. The PET images were averaged for all participants experiencing a given emotion, and activity during the neutral state was subtracted from activity during the emotion. Results showed that the activity was altered in many brain regions during emotional experience, and they were accompanied by significantly different patterns of brain activity. There is no simple relation between a specific emotion and change activity of a brain. Each emotion involves different differential patterns of activation across a network of brain regions associated with emotion. For example, activity of the cingulate cortex is altered in sadness, happiness, and anger. Another example is the left somatosensory cortex is activated in both anger and fear. And lastly, feeling of regret involves activation of the amygdala. In neural circuitry, hormones, and synaptic transmitters mediate violence and aggression. It says that violence, assaults, and homicide exact a high toll in many human societies. Many different approaches have been used to investigate the physiological, anthropological, and biological dimensions of aggression. In What is Aggression? It says that aggression has many different meanings. Intermale aggression is aggression between males of the same species. It is observed in most vertebrates, and in many male species must fight one another to gain access to mates. Men don't generally have to get mates, but they are physically aggressive than women. Males are 5 times as likely as females to be arrested on charges of murder in the United States. Aggressive behavior between boys is evident earlier in the form of vigorous and destructive play behavior. Sex hormones play a role in aggression due to the differences between males and females. In androgens seem to increase aggression, it says that the same testosterone that prepares males for reproduction will also make them more aggressive because aggressive behavior in males is adapted to getting food and mates in many species. For example, at sexual maturity, when levels of circulating androgens such as testosterone rise, intermale aggression markedly increases in many species. In seasonally breeding species like birds and primates, male aggression changes in sync with changes in testosterone across the seasons. In spotted hyenas, the females are more aggressive than the males, and the more androgen a female is exposed to before birth, the more aggressive she will be growing up. In mice and rats, decreasing circulating androgens by castration usually reduces intermale aggressive behavior. Treating castrated males with testosterone restores fighting behavior. Fruit flies don't make testosterone, but the same genes that trigger male courtship and copulatory behavior also promote intermale aggression. The relationship between testosterone and aggression in humans is not as clear-cut. Adults treated with testosterone do not increase their aggression, and young men going through puberty experience a suddenly large increase in circulating testosterone and do not show a correlation of aggressive behavior. Some studies have shown a correlation between testosterone and feelings of hostility. Non-aggressive tendencies in males are associated with satisfaction, family functioning, and with lower levels of serum testosterone. In female convicts, testosterone concentrations are highest in women convicted of unprovoked violence and lowest among women convicted of defensive violent crimes. 2 variables complicate the correlation between testosterone and aggression. One is experience can affect testosterone levels. For example, in mice and monkeys, the loser in aggressive encounters shows reduced androgen levels. This means that testosterone can sometimes be a result rather than a cause of aggressive behavior. Another example in men is testosterone levels rise in winners and fall in losers after competitions of a range of activities. It can also be shown in fans of sports teams or even political races. Another variable is dominance. For example, women treated with testosterone reportedly spend more time staring into the eyes of threatening faces, which is a socially dominant behavior. Testosterone models should be associated with behaviors that confer or protect the individual's social status. Surgical or chemical castration may reduce violence in some sex offenders, but the main effect is a reduction in sexual motivation more than a direct effect on aggression. And aggression has several neurochemical correlations. It says that activity in several neurotransmitter systems modulates aggressive behavior in various animals, including humans. Studies show a negative correlation between brain serotonin activity and aggression. For example, Higley observed aggressive behavior and fight injuries in monkeys from a large free-ranging colony to rank the animals from least to most aggressive. Researchers gauged serotonin activity by measuring a serotonin metabolite, 5-HIAA, and cerebrospinal fluid. The most aggressive monkeys had the lowest serotonin metabolites, which suggested they had the least serotonin being released at synapses in the brain. Serotonin is not the only neurotransmitter involved in aggression. For example, the balance between the inhibitory neurotransmitter GABA and the excitatory neurotransmitter glutamate appears to be important in aggressive responses to stimuli. It also says that aggression is regulated by many systems. The ventromedial hypothalamus is a hypothalamic region involved in eating, sexual, and aggressive behaviors. It serves as a switch to trigger aggression. When experimenters optogenetically stimulated neurons in the VMH in male mice, the animals would stop mounting a receptive female and begin attacking her. When experimenters optogenetically inhibited VMH activity, it reduced the likelihood that the subject wouldn't attack a male. In the biopsychology of human violence is a topic of controversy. It says that a controversy surrounds the idea that some forms of intense human violence are derived from temporal lobe disorders. Aggression is sometimes the main symptom in patients with temporal lobe seizures. A significant percentage of people who have been arrested for violent crimes have abnormal EEGs or other forms of neuropathology associated with temporal lobe dysfunction. Emotional discontrol system is a conditioning consisting of temporal lobe disorders that may underlie some forms of human violence. Psychopaths are an individual incapable of experiencing remorse and sometimes commit very violent acts. They do not react as negatively to words about violence, and they show little autonomic response to cues that have been associated with aversive stimuli during fear conditioning. Image studying showed that they have reduced activity in the prefrontal cortex and the smaller prefrontal cortex. And stress activates many bodily responses. It defines stress as any circumstance that upsets homeostatic balance. And the stress response has multiple stages. It defines the alarm reaction as the initial response to stress. The hypothalamus activates the sympathetic nervous system to ready the body for action, or otherwise known as the fight or flight. This includes sympathetic stimulation of the core adrenal gland, which is called the adrenal medulla. The adrenal medulla is the inner core of the adrenal gland, which secretes epinephrine and norepinephrine. Norepinephrine and epinephrine, or noradrenaline and adrenaline, are hormones secreted by the adrenal medulla under the control of the sympathetic nervous system, which prepares the body for action. These hormones act on many different parts of the body and boost heart rate, breathing, and other physiological processes that prepare the body for action. The hypothalamus stimulates the anterior pituitary to release a hormone that drives the outer layer of the adrenal gland, the adrenal cortex, to release adrenal steroids such as cortisol. Adrenal steroids are steroid hormones secreted by the adrenal cortex, including glucocorticoids such as cortisol and mineralocorticoids such as aldosterone. Cortisol is a glucocorticoid stress hormone of the adrenal cortex. And an example is Norwegian Army training during parachute training. The anterior pituitary released enhanced levels of hormones, and both the sympathetic and parasympathetic systems were activated. Cortisol levels were elevated in the blood before each jump head first, until the jumps were more and more successful and the pituitary response declined. Epinephrine and norepinephrine were also elevated before the first jumps, but eventually they returned to normal. Testosterone had the opposite pattern, falling far below normal levels on the first day of training, but returning to normal as the jumps became more routine. Once the jumps were mastered, they were no longer hormonal responses. Another example is factory work. The shorter the work cycle, the more frequently the person has to repeat the same operations and the higher levels of epinephrine. Another example is a train ride. The longer the ride and the more crowded the train, the greater hormonal response.